Hi, everyone. Welcome back to At the Bench, the physician scientist segment of the Incubator podcast. We're delighted to have you. Today, as in always, I'm Betsy Crouch. I'm a neonatal physician scientist at UCSF, and I'm joined here today by my co host, Dr. David McCulley, and by our esteemed guest, who we're delighted to talk to from across the pond, Dr. Eleanor Molloy. David, would you like to introduce yourself and get us started?
Sure. Thank you very much, Betsy. Yes, again, it's our honor to be able to host these podcasts and to try to give some insight into what it takes to become an outstanding neonatal physician scientist. And I am a neonatologist and a basic developmental biologist here in California at University of California in San Diego. I study lung development, and again, I'm thrilled to be able to talk with the guests that we have been able to invite. So today, I'm very excited to be able to introduce Dr. Eleanor Molloy, who is a neonatologist and physician scientist in Ireland. We have been collaborating together on a meeting that's going to be happening for the first time coming up in February with a joint collaboration between the European Society for Pediatric Research and the Society for Pediatric Research in the US. She is an outstanding advocate for physician scientists, and we're just so happy to have you join us on the show today, Eleanor. Would you mind just giving a quick introduction into who you are, and we'll follow up with questions about what's motivating you and the work you do?
Thank you. So thank you so much for inviting me. I'm really honored. I'm a huge fan of the show and your podcast. My name is Eleanor Molloy. As I said, I'm a neonatologist based in Dublin in Ireland. And I'm also a clinician scientist, and I'm very interested in immunology of everything to do with babies.
Yeah, thank you very much for that introduction. There are so many fun topics to discuss today. I think um, the breadth of your research is really exciting. Maybe we'll start just by asking you how you got into neonatology. I saw that you did fellowship at Rainbow Babies in Cleveland, and I'm curious, how you arrived there, and then how you managed to get yourself back to Ireland.
Great question. I sometimes wonder. Actually, my husband is a physician. He's a surgeon, and he got a fellowship in Cleveland, Ohio. And I went with him, and I was really lucky. I got to go to Rainbow Babies, which was the number one NICU in the States at the time. I was immediately given the textbook at interview. I was very impressed. And it was a wonderful time to be there. So I was there for a few years and got to work with Dr. Faneroff, Richard Martin, Maureen Hack, Cynthia Bear. It was amazing. And we had a great time. I also got to do a postdoc when I was there with Claire Dorshuk in neonatal inflammation. So it was a fantastic time. And I think what's really lovely about the American fellowships is that you are always their fellow. So many years later, I'm still invited a PAS to get to the gatherings. And when Cynthia Bear was looking for someone who was European to do the editor in chief with her for pediatric research, she contacted me. And that's how I've been in that journal, Pediatric Research, for the last decade because of my fellowship. And then going on from there, I suppose we always wanted to come back to Ireland, as many Irish people do. And I was lucky enough to get a job. And my first research fellow was Professor Afifa El-Kafashi, who I think know about and has been part of the show before. And he was a fantastic thesis student. And after him, then we just continued. But I didn't have an academic position for about 5 years. And then about 10 years ago, I got a chair position, which was fantastic. And then I could have my own lab. But my supervisor was amazing. He was a scientist who let me bring my PhD students to his lab for many years until I got my own lab. So that really, really helped. Um, and now we're really lucky. We have two postdocs and 5 to 8 PhD students who are all clinicians all the time. So it's great to be able to have that opportunity.
Eleanor, can I back up a little bit and just talk about how you got interested in neonatal inflammation and immunity and just how your research program kind of got started? I mean, you had some leaders in the field in multiple different aspects of neonatology, and I was wondering what it was about inflammation that was inspiring to you or interesting or something that you thought you could pursue.
Oh, no, that's great. I think what happened is, again, because I had finished my Irish training in neonatology, I had a little bit of a hiatus time where my husband was not going to go to America for a couple of years. And at that time in surgical training, everyone did a PhD, but you didn't in pediatrics. So I was very lucky that his mentor, research wise, became my mentor. And I was supposed to go to his lab to learn how to do a couple of techniques before I went somewhere else to do a PhD or a thesis. But actually, I ended up getting a grant and staying with him, and he was a neutrophil biologist. So even though he is now one of Bill Watson, who's now an expert prostate cancer, his original love had been neutrophil biology, and so that's how I started. And they were very kind to me, letting me do neonatology research in their prostate cancer lab. And then, and I didn't know anything about research at the time. I was probably kind of quite a senior fellow at that stage, but I really got interested in it. And as we were saying earlier, David, it really made me ask questions about everything in the clinical realm because I realized that nothing is perfect.
Yeah, I think that's one thing that is so helpful to learn about from science generally is it teaches critical thinking. So just the way that we approach questions in our research experiments and in the lab and in general like trickles over into everything that we think about. Like we don't take for granted that something that someone says is really based in any sort of evidence or has a fact base behind it. We really try to find that evidence and think about it critically and then, build supporting data that helps us come to a conclusion. So I think that that's really exciting to hear. And also just the point that you made about the opportunity to do research that was related to neonatology and neutrophil biology in a lab that was doing something entirely different because you can learn from people who are doing great science. It doesn't have to be exactly in the thing that you want to do related to neonatology. You can learn how to do science from somebody else who's using the same techniques, but in a totally different field. I think that's interesting for people to know more about too.
Absolutely. I think that as if you can collaborate as widely as possible, it's really good. And of course, then it's people you become friends with that you want to do research with and you meet at meetings. And I think you've been talking to other people about advice they give. And I think it's just to take all those opportunities and just really kind of see what they're doing. And I think as well in neonatology, we end up having to look at a lot of adult literature and adult research because that's probably where there's more funding anyway. Um, and it's always extrapolated, but we can learn things in both directions. So I think it's great to meet as many scientists as you can.
Do you have a particular story about, something that you you encountered in in the prostate either in the prostate cancer lab, which, made me giggle, the I was the neonatologist moonlighting in the prostate cancer lab, um, but, something from that particular experience or, just in general, as you mentioned, that there was, an idea or a trial or a hypothesis from the adult literature that inspired you?
Well, it's no, it's really interesting. It was a big joke that the only thing that neonates, the only organ that they probably didn't use or worry about was their prostate. But they did do a lot of renal dysfunction in the lab, so that was good. So, and you could see sometimes the urology experts who came in to hear about prostate research would be a little bit sad when it was me talking about babies. Um, but I actually was, what I did find though, and I think we find this all the time in research, is my best publications, as in the highest ranking that came out of my thesis, were all my adult controls. So looking at gender differences in adults' immune function got me much better publications than all those nights collecting cord blood in the middle of the night from patients with neonatal encephalopathy that took me 3 years to gather. It was that I was determined to get published, but the adult stuff was so easy. So I do think, but I think what was brilliant about that lab is my supervisor. He was very good at organization. So it helped me run a lab afterwards because I just copied what he had done. The regular meetings, the way he structured the lab, the way he organized things was excellent. That really helped.
Oh, tell us more about that. I mean, I think, we have a broad audience, which is beautiful, and, I really enjoy, now we run into people, across all areas of neonatology who have said, "Oh, I listened to your podcast," which I enjoy, and I really like getting the feedback. But I think there's a group of us who are early, um, principal investigators who could really benefit from more tips on expert organization in the lab.
Now, I can't say that I have expert organization in the lab, but we would meet very regularly. And I suppose what we've noticed recently since COVID is trying to physically meet so that we're actually in the same place as much as possible, because we're finding that between collecting patient samples, working in the hospitals, working as teachers, which most of our PhD students do as well, just to fund themselves, we're never in the same place. So I would actually have to say getting the community and meeting each other, even if your projects are completely diverse, is really important as much as you can. And then I suppose just making sure that we schedule individual time. I used to meet everybody once a week. Now we're they're more independent and they don't necessarily need that. So at least twice every 2 weeks with everyone individually. And then we just share the group. And what happens in our lab is someone could be working on something completely epidemiological, but they'll know all about the basic science and back and forth. So we do, nearly everyone is on everybody else's research. So I think that's really nice. And again, we have, because I'm chair of pediatrics and I teach pediatrics rather than just neonatology, we have people in our lab who are doing allergy, neurology, endocrinology, and so on. So it's a really nice mix. Of course, we try and always bring it back to neonatology, in some form. But yeah, no, I think it's kind of really getting everybody together and socializing is very important, because it is so easy to do Zoom, isn't it, and not meet.
It is, but I agree the connection that you have with in-person meetings is so much greater, and, it's much easier to be able to, I think, make progress when you're sitting together rather than because it's so easy to be distracted. Like, it feels like you're meeting when you're talking by Zoom, but there's so many other things that are kind of going on, and when you're actually sitting face to face or as a group, you can see everybody's engaged and the connection is stronger, and I think people's, like, critical thinking is a little bit more focused when you're in that kind of a meeting instead. So, yeah, that's good advice for sure.
We've started to do this thing where when I get asked to review a paper, we've started to do a journal club on it, where we usually meet in the afternoon. I bring snacks, as you were alluding to earlier, plus minus wine, and, we sit and we read the paper like a journal club, where each person has to go around and talk about each figure, and the engagement, as you were alluding to earlier, David, has been just phenomenal, and I think I feel, enthusiastic about it because the review that I'm then providing is such high quality, because I've actually had, 5 people from my group take a look at it, and different people, pick up on different things. My technician, my lab manager, who's, the most junior person in the lab recently, um, found, basically some evidence from a paper that we were reviewing that could indicate research misconduct. And it was him, It was, like, the most junior person in the lab who was just kinda Googling around the internet and, found Elizabeth Bick's, previous post on this group, and then was concerned enough to look more carefully at the data that was being presented. And I thought, like, because of this practice and this rigor, I feel like I'm doing a much better service for science, and, I don't know, that was really exciting, and, as you're both talking about, it's because we sit in person and we have the manuscript in front of us, and we spend, like, 60 to 90 minutes just looking at this manuscript with 5 to 8 sets of eyes. So, I think there is something that has become really precious about those in-person interactions and the quality that sometimes is missed when we're all, transactional and efficient, which there's a place for that too.
Well, Betsy, I think you've designed a new way of reviewing papers with wine and snacks that I really like, because I think, as you know, in publishing, the hardest thing is to get people to review papers. And doing in that fun way is really important. And then how can you change clinical practice if you can't read papers and you don't understand the science? So I think that's really cool. You have to kind of coin a phrase or call it something, the crouch review or something like that. And
Yeah.
we do like the shut up and write movement that we could do around the world. But that's something, and but I do think that doing it in person is brilliant. And you're
Yeah.
right, is often I find the medical students, you learn so much because they're looking at it with a different lens and probably in more
Mhm.
detail. and not glossing over
Mhm.
the things I'm glossing over. So I think having everybody is really useful. And we also have our parent representatives because we are really lucky. We've got PPI representation in our lab. So actually, our PPI representative picks out things all the time for us and is on our papers, and so it's a really nice perspective. But if I now I'm going to have to bring wine and snacks um, for all future meetings if they hear this
Well, I mean,
podcast.
it's Dublin, maybe you no, I had a lot of Guinness when
Yeah,
I was there, which it's tastier in Ireland than it is in the US, I'll say.
it is better.
Well, this is where I admit I'm not a Guinness drinker, but I hear the same.
Eleanor, you said you have a parent representative in your research group. Can
Yeah.
you just talk about that a little bit more? I think that's something we haven't talked about in
Yeah,
this
my mom
podcast
was
before.
along with that a little bit too. I mean, and I will say I'm leaving today to go to a meeting that's on a genetic condition that is run by a parent and a physician, and it's all patients and families and clinicians and scientists at the same meeting. So I'm a huge fan of this, and I hadn't even thought about a parent representative in my lab. So please tell us what we're missing.
Oh, no, I think it's, I think in Europe as well, our granting bodies have kind of insisted that you do have family members and parent groups from the inception of the idea the whole way through, and you have to show involvement. And so sometimes it always did in the past feel a little coercive. You go to somebody who you know and you say, "Can you help?" But you do feel that it's not fair. So we have tried to go more through parent organizations. But we were really lucky enough when we had a PhD program to have someone who was our coordinator who happened to be a parent. And she has changed everything in our group about how we write, how we think. And then we have other parent organizations as well that we've been involved in. And of course, we were talking a little bit about necrotizing enterocolitis. And I think the Next Society is an incredible example. That meeting is wonderful. And Jen Canavasser, with having inspired everybody at that meeting as a parent to start an organization like that that's so successful. And at that meeting, at every session, a parent talks about their experience, and it brings everyone back to what's important. It's a really nice. But I think, yeah, the more and more um, families are involved across lots of our areas of research, so it's great. Again, I do worry about taking too much time from people's lives when they're extra busy because they have a child who's been unwell. So then what we do is that's why we try and have a parent organization. So then different people can substitute for each other. So you have
Mhm.
someone in the group all the time. And then what we've done before with some of our PhD programs, which we try to do, is always have someone from the parent organization who's matched with the PhD student and PI. So they have their own kind of relationship to go more in depth into the project. Um, for example, if we had an EEG project or an MRI project, we'd have a parent completely focused on that, from everything from consent to what's useful to doing infographics and so on. So it's really nice. Um, and we have had interviews as well when we have parents there and they say why they're at the interview, and everyone does have a little cry. I have to say
Yes.
it's very emotional, but it lets the interviewee then tell what motivated them. So sometimes they'll tell something personal about why they're motivated for the job in a way that you can't really ask in an interview. So I think it is wonderful uh, to have that ability to have families involved.
Yes, it's so inspiring. I mean, the stories are very emotional, as you said, but I feel a sense of connection, and then also just the being motivated by what's motivated them, the questions that are they think are important, being directly involved and having a very powerful story. It's very compelling. And so then, I mean, we're talking about trying to attract people who are coming up into doing physician scientist work. And I think that is really helpful to be able to demonstrate it. Here we have this collaboration between this highly skilled researcher and also this parent advocate who has had a child with this disease, and we want to know more about the underlying basis of it, or we have this question that we've worked on together. I think that's such a compelling, just model for being able to, inspire, like, the next generation of researchers, but also, like, to just make progress generally. So that's outstanding. I have to say we don't in my research in CDH, we engage with families who have babies with CDH because we're collecting human samples. But for the most part, like the things that we are working on, like how does this gene function in directing diaphragm development or lung development, I think the parents are often interested in, can you identify an underlying cause or interested in the genetic mechanism, but like the nitty gritty of like how does this cell develop or things like that, I don't know. I'm sure they would be interested to know more about it though. Do
I
you
think
involve?
oh, sorry.
Yeah.
Sorry, David, you go ahead.
No, I was gonna ask Betsy if that's something that she's encountered in her own research, but
Well, I wanna know what Eleanor has to say because
I did
that
too.
yeah, and that's exactly what was on my mind because I think we're evolving from the idea that we, um, I don't know, do research in parallel with families to the actual
Yeah.
integration, which is what it sounds like Eleanor has, uh, envisioned well in the integration of somebody actually in the lab, um
Yeah.
but I'm curious, like, if there's an example because I think the other thing I'm going to this meeting today on 16p deletion syndrome, and we have a grant from the California Institute of Regenerative Medicine to understand the metabolic implications of vascular cells in this condition. And, Dr. Frankie Herrera, who's the woman who runs this organization, said metabolic implications of the disease can be a big factor in the lives of these individuals and children. So she said, I think you need to get perspective from the community on what it's going to mean to potentially intervene on metabolism in the setting of disease where metabolism is actually something that impacts their lives. Which I think was a brilliant idea and truthfully something that we were thinking about already. There was a number of physicians and physician scientists amongst the group who are envisioning the grant. But that's one of the goals of my presentation is to ask the community, how do you feel about, metabolic interventions for this disease? What are your concerns? What are your insights? And so I think we're moving to a place where where the families and the implications of the disease, we're starting to get a fuller picture, I think, of potential hypotheses that will impact back our experiments in the lab. Eleanor, do you have examples of this?
Oh, no, I think that's amazing. I mean, again, I'm thinking of Betsy Pylon, who is in Hope for
Mm,
HIE, and she
yeah.
is very, and she would be a
Mm,
huge parent advocate. And I think as well of Lily Collison in Cerebral Palsy Foundation, and they've just brought loads of energy, I think, to all the research. But they would be, again, David, I feel very interested in basic science and mechanisms and what's going on. But I do find then as well, we are kind of more inspired to do things like do clinical pathways in parallel as well. So even though we might be doing a lot of basic science in the lab, everyone does things like a definition. We did one recently of neonatal encephalopathy, or we do a practice guideline, or we look at what's the evidence for changing clinical practice in the different areas. So we would do them at the same time, which is
wi
nice for everyone who's a clinician anyway.
within your research group, you're doing that.
We would do that within our research group.
Yeah, that is great. I
Yeah.
think that makes it so much more translational when
Yes.
you may have a very focused basic science interest, but you're also
Yeah.
constantly thinking about how can we, inform the clinicians who are working in the hospital?
Mm.
How does our work relate to that? And if it's broad, that's great. If it's very focused, that's also great. I just think that interaction, that constant interplay between basic researchers and clinicians is great, but also just the same type of critical thinking that you applied to a basic science project and then extrapolating that to developing a comprehensive guideline is very important as well. And then having the parent perspective on both of them, I think, is also really exciting.
Yeah, because I'm lucky enough as well to work with Mandy Daly in Ireland, and she's a parent representative who has written so many grants and been on so many papers that she's a wonderful advisor actually about how to write European grants because she's just become so involved. So we are actually lucky that we have a large kind of group to call on in different areas of research. So it's no, it's fantastic. And I think the big thing as well is just making sure that the parents are on the paper as well.
For sure.
Even yeah, even if the paper is in an area, as you said, that might be very detailed, I think it's really important that they probably had to listen to us talking so much, but I think it would be, that's really important so that It's not like the scientists are doing one bit that everybody's involved in every bit.
Can you talk about just for a second longer? You said the granting agencies in Europe are really motivated to have parent engagement or family patient engagement, I guess, in research. And so, in order to apply for funding, you need to have a patient representative as part of your application. Is that right, or how does it work?
Absolutely. We've had, usually we'd have parents as co-applicants as well as collaborators.
Mm,
And then in previous projects, as I said, we'd actually always paired a parent into each PhD program or PhD project. And then it would be just showing evidence that at every single part of the research planning, execution, and publication and dissemination, the families and parents are involved in that. And that would be kind of a core part of every grant. And actually, our granting body in Ireland then gets every grant reviewed by a parent representative. So you'll have that perspective. And did you really explain it properly? So it might not be a family member in the same area that you're writing the grant in, but they might kind of question it. And so yeah, so it's actually, so it is very, very important part of the project. I have to say the most stressful grant I ever wrote was one where all the parents wrote it and they wrote their segments. You know, when you ask someone to say, what is your contribution? And they wrote about their own child and their own experience.
oh my gosh.
And when I read the grant, it was like, oh my goodness, this is the most amazing grant. And I said, how? What are we going to say if we don't get it? And we didn't think we were
Yeah.
going to get the grant. So I think that's the only pressure is just saying to people's expectations, look, we might not get this, but yeah, but yeah, so I think it's and we try to do in the journal as well, parent reflections and family reflections,
Yep.
and more we're noticing people are including those in grant applications.
Hmm.
You know, where we ask parents to reflect on what will be useful. Say, for example, in necrotizing enterocolitis, where's the research? Where does it need to lead? Try to have that perspective.
So it sounds like inflammation was something that was interesting to you, and then you talked about early on you were interested in sex differences.
Hmm.
Um, can you talk about that? It seemed like it focused on neonatal encephalopathy, so that seemed to be like where you were kind of interested to get started, but now it's expanded widely. So I'm just curious to hear a little bit about how you developed that interest and like what are the things that you're trying to learn more about?
Well, I have to say, yes, my thesis was core based on neonatal encephalopathy, but then it did have to expand just because patient numbers, and it took
Mm,
me 3 years to recruit my patient numbers I needed for that. Um, and I'm still really interested in neonatal encephalopathy, and that is a core thing. However, what I intend to do with the research is I do have to follow the money in the grants, is part one
mm-hmm,
that we all have to do. And then as well, I kind of follow who our PhD students are and what their interests are in.
mm,
So that does end up meaning that you go in slightly different directions. And then as neonatologists, of course, we're interested in all organs,
yes.
which makes it a little bit harder to focus. So I think that so sometimes what we'll do is we'll go with what the PhD student is interested in, and then we'll try to see how can we do multi-organ. What I really like about inflammation is it affects every organ, it affects every baby. It is something we could modify and change. But I worry about trying to give medications to children without actually understanding what's happening in their inflammatory system. And I think there was a group that was talking about sepsis recently, and we were saying how a lot of neonatal immunology is based on cord blood, which is completely different to actual babies. And we
Mm-hmm.
want to kind of rewrite the whole world of neonatal immunology. The other thing I like about it is that if you have an abnormal dysregulated immune response at birth, it does seem to persist, irrespective of whether you have NEC, your preterm, you have neonatal encephalopathy, your cerebral
Mhm.
palsy, it just, or Down syndrome. That when you check children later, and we're trying to do more adult work now as well, this seems to continue and may contribute to long term issues. So, what we'd like to do is see what interventions you can use. And then our kind of ideas in the lab is we'll take blood from a baby and we might look at what's been done in animal models. And then we will treat that blood, we'll try and stimulate it and see can we decrease these abnormal inflammatory responses or augment them, depending what the case may be, in the lab, and just see what doses work and then see can you extrapolate that with animal models into clinical trials. So I think we kind of feel that we're that human translational piece in between. Um, and then I was saying it does depend on the PhD student and what area they're interested in. We do tend to adapt. And because inflammation affects every organ, and we're neonatologists, we like every organ, we
Uh-huh.
kind of try to put everything together. And it is sometimes hard because we don't really have a home society for neonatology where everyone sits in the room and talks about inflammasome inhibitors across lung and heart and kidneys. We're all little bit separate silos, which is probably a necessity too. So I like inflammation because it mixes that all together. And we see similar kind of patterns across different groups of patients. Um, but so going back to neonatal encephalopathy, that is nearly in some ways more straightforward because they all have a massive hyperinflammatory immune response. So you could envisage that you could use the same type of drugs and interventions in that group. Whereas I think with neonatal sepsis, our babies are dynamically changing so much that you have to be careful about knowing where they are and what drugs would have to be tailored for them and individualizing it and personalizing it to that patient. So I think that's very interesting.
You mean at their developmental stage, Eleanor?
I think tailoring, yeah, and through childhood, and not just. I know that we say, for example, in neonatal encephalopathy, use therapeutic hypothermia as a baby, and then we don't think about treatments later in childhood. But I think thinking about those treatments and then looking at all the other organs, like have you been checking their blood pressure throughout childhood? What's their kidney function like?
Hmm.
Um, how's everything else getting on? And I think that's something that we don't always think about all the organs.
One thing that seems to translate through all of your work is just looking for differences between males and females and looking for differences, different gestational ages. And I was just wondering, I mean, aside from just demonstrating that males and females are different and biologically different and understanding that might help us to be able to treat them more effectively, how else are you thinking about it? Like, are you trying to understand mechanisms of disease that are unique to males and females, or what's your thinking about that?
I know it's a really good point, Dave, because when we do this, yes, first we're saying everyone's different, but then what can you do with that information?
Yeah.
And I know in the past there's been some clinical trials where they've used estrogen analogs only in preterm baby girls, shown improvements in lung function and development, but in small studies. And again, you can't really give estrogen analogs to boys at the moment. So
Hmm.
I suppose I'm kind of wondering, and this again more as epidemiology is should we be using different doses of steroids in baby boys, especially twin baby boys? Should we be tailoring that more by sex because um, girls are probably making more estrogen and protecting themselves better? So I do think there's that element. And we found that in studies where we added estrogen and progesterone, we could change to blood, that is, not to babies, we could change their inflammatory profile, um, and we found that as well in adult human blood, you could make a male neutrophil actually like a female neutrophil if you treat it with estrogen. So I think there probably is a space for medications that don't have too much estrogenic effects, but have steroid effects that might be beneficial. So I'm looking at boys and thinking about how we measure steroids. Again, steroids are hard to measure, as you all know. But I think now with really good advances in mass spec techniques, we can probably measure it more accurately and see could we tailor to that specific child.
I think that is just such an important point to highlight. I mean, this is a topic that's received a lot of attention recently in the US. And it's something that it's just important to demonstrate that there's important biological differences. And without understanding them, we're just losing many opportunities to be able to better treat patients. So I think it's just one thing to just keep focused on is just like, how does doing this research impact our understanding of the mechanism of disease, but also like why would a one treatment that we use all the time potentially be risky in one subset of patients because of their biologic background, their sex?
I think it's so interesting moving from protocolization, which certainly is one way to improve our care, right? Is to make sure that everybody knows about, certain quality metrics and that we have, a standard way that we treat every baby. But I think the next stage, right, is personalization beyond the protocolization.
Hmm.
And yeah, and I think biomarkers are going to be an important part of that. So um I wanted to follow up on a different, scientific thread that you mentioned earlier, which is the fact that cord blood is not representative of the baby blood. Could you talk about that a little bit more? Because I think like everybody, we're interested in the immune cell. effects on different organs, and our favorite organ is the brain. And so it would be helpful for me personally, and I'm sure for our listeners, to hear more about the differences between cord blood and the baby's immune system.
So there's a lovely paper by Peter Broden where he looked at in absolute detail at cord blood and then neonatal blood all the way through childhood, looking at every different protein and immune function, and just showed large differences. But it's cord blood is absolutely wonderful in research if you want a large volume of blood and you have to do a big experiment. But we find that you would have to give a lot of stimulation to the cord blood to get a response. So sometimes we actually have to make it hypoxic. We have to give endotoxin about 3 times, and then it responds. And so I think that has been where everyone has always thought that babies don't have a very immature or abnormal immune response. So we got a big fright when we started doing neonatal controls from babies having blood for other reasons. We found they actually produce more cytokines than adults do, and they respond really well to endotoxin stimulus. And we were kind of worried our controls were wrong, but we noticed a lot of other people were finding this too. So I think that babies actually can respond really well, but they are adapted to not be rejected by their mother and vice versa when they're in utero. So they have a slightly altered immune response, but it's not that it's necessarily deficient. And I suppose that's why later on we see such a massive inflammatory response with necrotizing enterocolitis. I also wonder about pulmonary hypertension as an immune response early on as well, that there's an element of that. Now, David is the expert on this area, but I think there's lots of things that probably have an inflammatory element to them.
Ooh, tell us more, David or Eleanor.
Yeah.
I was just thinking, just because you talked about just how inflammation and the immune system affect all organs, and I was kind of then wondering, in addition to thinking about the patients as individuals and their immune system making them unique, I wondered if you have found instances where in one organ, if you modulate the immune system, you have one effect, but you might have the opposite impact on another organ. So, I mean, pulmonary hypertension is a good example where, um, sort of inducing a fetal program may be poor, may have a poor outcome, but being able to preserve development, say in the brain or in another organ in a preterm infant that has an inflammatory reaction to NAC or sepsis, may be beneficial because it allows for continued development. I don't know if you've found that or if you're thinking about that. It just seems like being able to target individual cells and different organs to modulate their immune response might be important, but I don't know.
Now, I think you're definitely getting more advanced than I am because I'm very simplistic and have and only work with blood, which probably goes to every organ. So I probably look more at just immune cells. But you brought up something really important, David, is about um, the immune system. Nearly every cell in the body can respond in some immunological way. So it doesn't have to be just monocytes, neutrophils, and lymphocytes, which is what
Hmm.
I work with. So I do think that's really important, but very hard to measure in human beings. But I do think more and more, especially when we think about necrotizing enterocolitis and the gut immune system, is really, really important. But I don't know completely the answer to that because I'm assuming that the blood, where we're taking our samples, it goes to the entire body.
Hmm.
Um, and I suppose what's handy about that is you can see the cytokines in the systemic circulation, and you know they're going to affect brain and kidneys and heart. Um, and kind of protecting the babies against that storm. But you brought up another important thing is I suppose you have to modulate it because you can't just block it because then they're going to get sepsis and it's all going to be, it's not going to work out well. So how do you actually modulate the immune response and just get it to the exactly right level? We're calling
Yes.
it the Goldilocks zone. Has to be just right. How do you get there?
It's right. I really love thinking about that. These things that are in biologic balance. You can't just think of it as an on or off. You have to think about it as like kind of a tuning. And I think that's just another level of complexity is like how do you find the appropriate tune? And also that it may be different from one
Yeah,
cell in one organ and its neighbor, even in the same organ. So it's very there's so many questions to ask. It's very
absolutely. And I
interesting.
think it's so dynamic that it might be different 1 hour later in the same baby. And I
Yes.
suppose because you study pulmonary hypertension, you see that all the time.
Hmm.
that, suddenly the therapy you're using, it might be a bit too successful.
Right, it's caused a problem in the out
Yeah.
in the just a few minutes later.
Yeah, and, on this topic, there's some emerging literature from Wendell Lin, who's a scientist at UCSF, where the idea is that you have immune cells that are synthetically engineered to be gated for a specific organ. So, so that the immune cells, when they enter a specific organ, then they encounter some endogenous antigen that then activates them to do a function that they otherwise are restricted or inhibited from doing. So, I like this topic a lot, and I think that we could um, learn and borrow from our, cell engineering friends and colleagues in terms of how to activate the immune system, specifically in an organ targeted way. But yeah, that's a very exciting opportunity.
We wanted to ask about was just that it seems like you have been incredibly successful in your ability to collaborate internationally. And, to people, I don't know how Betsy has done it, but for me, it's challenging to collaborate internationally. Like, I meet with people at meetings where, we're working in different countries, and it's interesting to be able to see, like, what you can do, say, in Germany or in Great Britain or in other places, but it's difficult to actually get grants together and things like that. Can you talk about your international collaboration and things that have worked well and challenges that you see?
Oh, no, I totally agree with you, David. I think it is really hard. What's handy is, I suppose, having worked in the journal, which is the official journal, say, for Society of Pediatric Research, European Society of Pediatric Research, and American Pediatric Society. Therefore, we're always trying to see how can we get those societies to collaborate and write papers together. So I think, and then in the European Society of Research, it's actually divided up into sections. So there will be a pulmonology section, a resuscitation section. We've got next special interest groups. We've got special groups on stem cells and so on. So what we often do is harness those groups to write papers, and then they will get their international community involved. And then you're focused on writing a paper together or doing a task. Because I do agree with you, we can have lots of meetings and talk about things, and sometimes getting grants together, they're not successful, and then that whole group falls apart. So it's trying to find tasks that you can do as a group, or even or going to a meeting like the next society together, or things like that that you have to organize. Um, because otherwise the group does kind of lose momentum, I think. Um, and I look at very successful groups in neonatology. I think the Neonatal Kidney Organization, the Next Society, of course, in cardiology and pulmonology. There are so many, a Newborn Brain Society, and that they all kind of work on tasks together and have subcommittees and groups. And I think that really helps. And then getting those groups together so that you can harness them to make networks probably is the easiest way to do it. And then you have to have someone with amazing energy, like I'm thinking of Mohammed El Deeb and the Newborn Brain Society, who made it all happen. And I think that's so sometimes I'll be sneaky and get into one of those groups so that we can do things together. But I think writing grants across continents is challenging.
I like your idea though, the just achievable tasks. Like, because if you start with like, we can write a paper together, we can write a
Yeah.
summary, or we can develop a guideline together.
Yes.
We can explore this topic together that we all have a shared interest in, and publish something that's meaningful and it's going to be cited widely because we have people who have been really dedicated to this topic and they have important insight in the field. I think that is an achievable thing, and then it engages people who to participate in a society. So you have like you've achieved a goal, you've published a paper related to this topic, and then they want to come to your meeting again the next time you meet. And so then it becomes iterative. But I think it's that next level of like, how do you then build funding mechanisms or take advantage of funding mechanisms to keep it going? I think I don't know how to do that yet, or I don't know.
I think I totally agree. I think that's a really difficult thing. I do think there's some sections in ESPR, like pulmonology and resuscitation, who've done that really successfully,
Mm.
and where they've started doing maybe uh, transnational questionnaires and working together on papers like that, and then have developed these into doing international clinical trials together, and have done them internationally, but maybe their basis was in these groups. And I suppose we're always trying to get the groups to kind of cross. the Atlantic and Pacific. So it's because and then of course it is part of the publishing thing is we think the messages are more powerful if they come internationally. So we would be really encouraging that.
So then let's go to that next. Just your role in pediatric research, and you alluded to how you got involved in that with through your connection with Cynthia Bearer, but um, just as a neonatal physician scientist and someone who is trying to foster the next generation of researchers very broadly, um, how do you see your role at pediatric research? What's exciting about it? Any messages for our audience that you think would be interesting?
Well, it is really fun, because you get to see everything that people are writing. And then,
Mm.
for example, now, David and Betsy, I can email you and say, "Oh, I'd really like you to write a paper on," and people might actually reply to you and do it. So
Yeah.
that's really nice. And I go to meetings and we kind of go around looking for people we want to write papers for us. So that actually is really nice. Um, and you gotta keep up to date with what's interesting, what's hot. Though pediatric research is across so many different specialties. I mean, I definitely am not an expert in endocrinology, so we're really looking with endocrine specialists and so on. So I do think for any young investigators, we have opportunities for young investigators, things like reviewing articles, getting your mentor to help you to do that. I would think, okay, I mean, Betsy's, okay, that might be more tame than what Betsy had described, which is more fun. But reviewing articles is really important because it helps you understand how to write one yourself. And I think we also have opportunities like with travel awards and so on. But then I would say just to be brave and go to your mentor and say, "Can I write something with you?" Is really helpful. And just to take every single opportunity to write something. I mean, what we've done as well is we've been brave when we did neonatal sepsis definitions. I went to all these famous people in pediatric and adult sepsis who I thought would definitely say no, and surprisingly came on steering committees and so on. So I think it's about just what's the worst thing that can happen? Is they just going to say no or they're too busy? But I think it's amazing how people will engage with you. So I would encourage people to kind of literally go and ask. Um, I definitely went to Richard Martin was our mentor in Cleveland and
Mhm.
just asked him, could I just write lots of papers? And he was brilliant and that really helped. So I suppose in the journal, I do think that, yeah, I mean, I'm going to say the boring thing, publish or perish. If you did that work, you've got to publish it. And then what's nice about having family engagement is you ask those parents to participate, and you got their consent and you went through ethics. So I think when you don't write it up, I do feel it's kind of nearly ethically not fair on the families who are involved in the
Yes.
study. So I do think and then it's how people can help you get through the last phases and getting your group together and maybe having writing groups so someone else could finish it off. Because I think that's the hardest bit, isn't it? So I would just encourage people then, if you feel you've got your paper and you can't bear to look at it anymore, is to find someone else who'll do it for you. And just do that last push. I think everybody knows what that is. But,
It's just interesting to hear how different people do it. I think everybody has a slightly different approach, and I think even for the same person, it might vary from time instance to instance. But I think just that there's a community of researchers who are all doing the same general idea, but finding things to motivate them. And I love the point that you made about how having parent engagement helps you feel motivated to get over the finish line because sometimes it is you feel like it's just insurmountable. You're not going to be able to get it together, but that is I could definitely see that that would make me want to get it published and just to feel like uh, to honor their participation.
yeah. I think you have to find any hooks that can make you feel guilty enough to get it published. Because I think we all know it is quite a long journey of persistence. Um, I think it's anything that you can use, someone making you feel guilty, whether it's your PI, supervisor, colleague. And that's why sometimes having other people on your paper and having writing groups really helps.
Yeah. It is a long journey, but a rewarding one. As we're getting toward the end of our interview, we like to finish on something fun. So we've learned a lot about the activities that different groups do. to bond or just to, add some lightness to the intensity of our scientific investigations. Is there something that you and your lab group enjoy doing or a tradition that you would tell us about?
I was asking them today in lab meeting what I was going to say for this question.
You knew this was
And
coming.
I knew it was coming, and I said, we have to sound like we're great fun. But I suppose we do go out quite a lot and we celebrate a lot because most when our fellows do leave after the PhD, they're probably going to a different country to do a fellowship. So we
Mm.
would have a lot of get togethers and we do spend time together. Um, we go for a hike every year, and that's
Oh, wow.
organized by our institute where we kind of all go with our dogs and other family members up a mountain, which is lovely near in a beautiful place in Ireland. And then one of our postdocs has organized escape rooms and things like that. So we do try to do other activities together as much as we can. In the journal, we are obsessed with dogs. I
Hmm.
have a disclosure there. So if anyone wants to send us papers about dogs, and we would
Inside
commonly
tip.
so our outlet is our kind of our family and our dogs and so on. But my only thing, I suppose, is how my work and life blend together. When I was going for my chair position, I would have been sitting outside football matches with my children. And I remember several nights as I was finishing presentations, loads of mums getting in the car with me and giving me career advice and changing my slides and doing things like that. So I think it's amazing how much support you can get from people looking and saying, "Oh, I don't like that slide," who might be in a completely different area of work to you. So I think we do all bring our work to our fun and vice versa.
Yeah, you can pick up good advice from anywhere, it seems, and that's really good to take advantage of it. And also, just good to hear because I feel like getting another person's perspective on what you're talking about or trying to learn about, and then present. If you can convey it to them, and it's not their field at all, I feel like you can get so much good insight and just do a better job of talking about your work. So that's great. Good
This is
example.
very California, but one of my PhD students, Cassie, recently, told me how she was networking and she's surfing recently, and I thought, "Well,
Oh,
that's a
wow.
cool one," but, very California that, in Ireland, you take hikes with your dogs, and in California, I imagine she's, like, on her board and riding a wave and talking to the person. She says, "I keep meeting all these scientists in the water." I said, "Oh,
Yeah.
well, it's productive, so good for you."
Now, sorry, that is definitely the coolest collaboration method that I've heard.
Okay.
The
Well,
surfing collaboration.
yeah, the surfing collaboration, so we'll start the new neonatology surfing society. There we go. We have ended this interview on the peak of inspiration.
Thank you so much, Eleanor. Yeah, that was outstanding. Really fantastic to talk with you. Thank you for taking the time.
Thank you, Professor Molloy, for a wild Irish ride, and, uh, and for really, I think, showing us a different way, a broader way, a more international way to think about the differences that we can make as neonatal physician scientists. So this has been a delight and really thought-provoking. Thank you for joining us today.
Thank you both. It was wonderful.
Perfect. Thank you so much.